Saturday Morning Physics is sponsored by the Physics Department of the University of Michigan, by the Dr. M. Lois Tiffany Endowment, and by gifts from friends of the program.
I will start with some brief historical comments on the history of some unpopular discoveries. Nicholas Copernicus, he published in 1543 a work known as De Revolutionibus, where he talked about our solar system as being heliocentric rather than Earth-centric. It was published in 1543, shortly after he died. It was actually written 13 years earlier. He just sort of sat on it because he knew it was going to be unpopular. And even before it was written, the idea had been circulating with him and with some of his colleagues as early as 1510. Galileo picked up on this and talked about it more in his 1632 book, Dialogues. And the following year, actually took less than a year for him to be sentenced to prison by the Inquisition. And interestingly, his book, Dialogues, stayed on the index of prohibited books until as recently as 1835. This is something published by the Roman Catholic Church. And so here we have, long after the fact, just a schematic describing some of the photographs taken by Voyager 1 spacecraft that showed, after all, yes, indeed, we are in a heliocentric universe. Each little square is a picture that the spacecraft has taken, and if you get enough of these over time, you can map out the orbits of the different planets. So, Copernicus was right. And it took about 325 years from the first publication by Copernicus to its acceptance as measured by being taken off the list of prohibited books. So this is very arbitrary and subjective, but I think it's at least a reasonable way to measure the time to social acceptance for heliocentrism, 325 years. So I want to take a look at a couple unpopular discoveries in the context of how long it took for them to be to be accepted widely. Girolamo Fracastoro is really the father of germ theory, and this is another unpopular discovery. Historically, this has been fought by organized religions very early on. He published a book in 1546 called On Contagion, and in this book, he put forth the heretical idea that diseases was caused by invisible to the naked eye particles or germs. And he said that different diseases were the result of different particles. And of course, this was unpopular because some in the church felt that diseases were something that were for divine power to either visit on people or to take away. So this was quite unpopular. He was a friend of Copernicus at the University of Padua in Italy. And he published this, and it didn't really have much of an impact at the time. Fast forwarding to the time of Robert Koch during the 19th century, we can see that this is when it really provided the proof for this unpopular discovery, that in fact diseases were caused by microbes. It just shows some anthrax bacterium, and his contribution was just in developing the stains. In part, his primary contribution was also in linking the presence of these bacteria to diseases, and doing this for a number of different diseases, including tuberculosis, cholera, anthrax. Where you found these microbes, you found the disease. In all cases where you found the disease, you found the microbes. You could introduce the microbes externally to either in cell culture or to somebody who is healthy and induce the disease. And so this is how we have Koch's postulates for linking particular diseases to particular germs or microbes. So in this case, we can say it's about 335 years to the acceptance of a very unpopular notion, that is germ theory, going from Fracastoro to the work of Robert Koch. So those are two unpopular discoveries, both taking over 300 years. Benoit de Maillet, he had this very long title, but maybe some of you had seen. In the title of his work is his own name backwards, de Maillet. And that he used the same literary artifice that Galileo did in putting all of his heretical ideas into the mouth of somebody else, his Indian philosopher. And he really did outline a theory of transformation. So I would like, given my arbitrary power to decide how long it took an idea to move towards acceptance, talk about Benoit de Maillet as one of the first persons to put down an evolutionary theory of sorts. And toward this end, I wanted just to read you one quote from his book. So this was written probably around the year 1710, something like that. So consider this quote in the context. And remember, it's always worth judging people, scientists, in the context of their own time, not in hindsight. So think of what he had to work with as far as evidence or prior conceptual ideas to work with. So here's the quote from him. "The transformation of a silkworm or of a caterpillar into a butterfly would be 1,000 times more difficult to believe than that of a fish into birds if this metamorphosis did not occur daily before our eyes. The seeds of these same fish transported into marshes may have also originated the first transmigration of the species from the marine to the continental environment. If 100 million individuals perished in contracting the habit, it's only necessary that 2 should succeed in order to start the species." So his discussion of mechanisms is, of course, laughable, but only in hindsight. So we can start there with de Maillet. Lamarck published a phylogenetic tree of sorts in 1809, and of course Charles Darwin in his book Origin of Species really provided the mechanism that convinced the scientific community that evolutionary history was real, had happened. We could see this was the mechanism for evolutionary change. Since Darwin's time, we know that there is plenty of time for evolution to happen, 4.5 billion years. We've identified the heritable material, DNA, and of course Darwin had no idea about this. And so if we do the same thing with this unpopular discovery, we can talk about it's really only been 288 years that have passed since de Maillet's publication up until 2006. So in the long view, we're not doing too badly. And you know, this is a tough one because arguably evolution is even more or is as unpopular or more so than any of these other previous discoveries because it's so deeply personal. It has to do with human origins and has to do with individual origins. And so it's actually not that surprising, at least to me, that there should be a great deal of cultural inertia against acceptance of this idea, which of course has been accepted by the scientific community since the mid-1800s. But that's another thing to convince society at large. So we're sort of on track there. And so what I want to do now is move to talking about some of the ways in which our understanding of evolution has directly benefited us. And this understanding of evolution may either be intentional and conscious of the details, or it may be just sort of a common sense notion of what really works over time. And I would maintain that humans have been using evolutionary phenomenon by selecting upon variation within wild species to come up with all of our plethora of domesticated crops and animals. So I have a series of drawings, for which I'd like to thank John Megahan for doing the original artwork on these, that talk about the history of domestication for some species. So here we have a gray wolf standing astride the world. It seems that gray wolves were first domesticated in Eastern Asia about 15,000 years ago. This is based upon molecular systematics work in which we find the greatest divergences between breeds of dogs that are native to Eastern Asia. The greatest divergences are found between those breeds of dogs. So at least that's our working supposition, is that the first domestication event was probably in Eastern Asia about 15,000 years ago, making use of the notion of a molecular clock, which has been talked about during early Saturday morning sessions. We have evidence of a human burial in what is today Israel of a domestic dog skeleton neatly tucked under the arm of an elderly female who's buried under a tomb of rocks. And this dates back 12,000 years ago. So there's archaeological evidence as well. We have a dog here in the audience this morning, Daphne, who is some evidence of humans selecting on dogs for all kinds of traits, and Daphne is being sort of trained and will be assessed for her aptitude for fetching things for people with low mobility, possibly for pulling wheelchairs, and just generally being of assistance to humans. And of course, these are familiar breeds. Here we've got King Charles Spaniel, been selected for small size and companionship, a work dog, the Australian sheepdog, a guard dog, the bull mastiff, hunting dogs, the Labrador retriever, work dogs, also the Siberian husky, and a greyhound selected just for the pure pleasure of speed and grace and the uncontrollable human urge to gamble. The similar kind of story with horses, although perhaps not quite so old. It seems, based on molecular evidence, there's been multiple incidences of domestication of horses from Przewalski's wild horse. The current evidence suggests it goes back at least 6,000 years ago. We know from archaeological evidence that horses were used in combat 4,000 years ago, as early as 2000 BCE, before the Common Era. Here is a phylogeny just showing where Przewalski's horse / domestic horse, that would all be the same species, fits in with some of its close relatives. Here's a donkey and zebras, our sister taxa here, and here's tapirs and rhinos. So out of wild stock, we have developed, as with the dog, all these different breeds of horses for different uses, for show, for fighting, for work. The Shetland ponies, many of which were born underground in coal mines, never saw the light of day, were hauling coal carts underground, one bred for speed and also gambling, the thoroughbred, and of course draft horses. Cattle, it's much the same story. Here's a drawing which may be familiar as a common cave drawing of an auroch. The last aurochs went extinct in what is now Poland in the early 17th century. And from these aurochs, humans have developed, through artificial selection on the variation that was present, selecting for particular traits that they thought would be useful, a whole variety of strains. Here is a Holstein selected for huge amounts of milk production, way beyond anything that would be seen in wild ungulates. Galician Blonde for meat production, Endama selected as a work animal, all-purpose work animal, also some milk production. The Dexter for relatively small size, bucking bulls, Ancol, and so on. And of course, at the same time, with the Endama, they've also have evolved some resistance to malaria parasites. And this is why it's not easy to take breeds of livestock that are bred in one region of the world and transplant them someplace else, because there are some adaptations to the local environment, and Endama have that in some immunity for some African parasites. Wild red jungle fowl would lay about 5 to 10 eggs a year. Laying chickens now will lay over 300 a year, all brought about by natural selection. Our first evidence for cultivation goes back at least 19,000 years, that humans have been selecting upon different kinds of these grasses as a food staple. This has been tremendously important in facilitating the spread of humans across the globe and the development of human culture, because we could store foods like this. The development of cabbage is a fantastic story. Here is a wild Brassica oleacea plant, the wild cabbage, and a line connecting it to each part of the plant, which has been the focus individually of humans selecting for particular products or traits, and these are all readily recognizable. Rutabaga, also from this part of the plant, mustard greens and kale from the leaves, head cabbage from the terminal bud, Brussels sprouts from the lateral buds, broccoli and cauliflower, which are really the same thing, from immature clusters of buds. And white and purple kohlrabi as well, which includes the leaves and part of this swollen hypocotyl. All of this developed by human beings from a single wild progenitor. And of course, without all this, we wouldn't have sauerkraut and other things like that. Coffee, the official beverage of Saturday morning physics. First evidence of cultivation is in the 6th century of the Common Era in this part of the world. And probably the berries were first eaten, but people couldn't help but notice the stimulatory effect that coffee would have. It was first brought to the New World in 1723 into the Caribbean. This has become an important part of human culture, you might say. especially within academia and many other. So there's been lots of artificial selection by humans on crops and plants. And even though people weren't aware of exactly how, well, people were aware of how evolution worked. You know, that progeny tended to resemble parents. And you just chose those parents that you wanted to contribute particularly to subsequent generations. And so really it's a common sense application. And the application of artificial selection continues when we make attenuated, that is weakened vaccines. The way this is done is simply by infecting and growing in an unnatural host species a particular virus, or letting it grow under extreme conditions, so you're selecting for particular mutants. And those mutants might be strong enough to survive and elicit an immune reaction from a host, but not strong enough, since it is weakened, to kill the host. So you infect and grow it in an unnatural host species, some of the new mutants survive, and then you can go through multiple bouts of this, and this is a way in which things like a polio virus has been developed, rubella virus, a measles virus, and there is still discussion of work on HIV viruses. This is also artificial selection. This would not happen if we did not understand something basic about evolutionary processes. Something that's perhaps less well known is our harnessing of evolutionary processes in something called directed evolution. And here we can really make designer molecules, so to speak. And here's just 6 steps in the ways we can make directed evolution to make particular molecules that are useful to us. First, we select a gene, and this is just being shown as a linear sequence. Let's take a single gene like that. Here's the DNA sequence represented by this line. You create a library of variants of this gene. So you've got mutations here, there, there, and there. You insert this gene library and a vector, something to carry it, like a plasmid, into bacteria, which produce variants of this enzyme that's encoded by this gene. You screen all of those different variants for the property of interest, ability to metabolize something, ability to grow in a particularly harsh environment, and you isolate those colonies that perform particularly well, and you repeat this process over and over times thousands of iterations. And you can really push molecules in the direction that you would like to do. And people are doing this. Another sort of evolutionary approach to the same sort of thing is known as DNA shuffling, where you would take the homologous gene, that is the same gene because of common descent, in 4 different species of organisms. You chop them up and allow them to reassociate randomly. Because these genes that encode proteins and the proteins have specific folding patterns, you need to choose genes that are homologous, that is, they are the same thing by common descent, because when they reassociate like this, you can at least hope to maintain that tertiary, that 3-dimensional structure. So you need to have a little bit of evolutionary information on these particular genes, and by doing the shuffling of chunks of these different genes, you can make a chimeric gene and then test that as well. So really, this is, natural selection in human hands at the molecular level. You can use this approach to enhance existing functions, things such as selecting for vaccine components that strengthen the immune response in humans. Okay, if a vaccine is weakened, well, maybe you want to tune it up and make it a little more powerful to elicit a stronger response from the immune system. You might select on proteins such as cytokines, which have a very important role in down-regulating autoimmune attack. That is, when your own immune system is attacking your own tissues. That would be a good thing to be able to down-regulate if possible. Autoimmune diseases include rheumatoid arthritis, multiple sclerosis, inflammatory bowel disease. You can also use this approach to select for enzymes conferring resistance to insect and fungal pathogens in crops. You can use directed evolution to try and develop new functions. Selection for RNA inhibitors of HIV replication. This is still a promising approach to dealing with HIV infections. Inhibit the replication mechanisms. Selection for enzymes which block cancerous cell growth. This would be a good thing to do. Selection for enzymes which degrade pollutants. In all of these ways, we can use evolutionary approaches and our increasingly sophisticated understanding of the processes of selection and what we need in terms of population variation to make these sorts of approaches work, and to target them to particular regions of particular genes. In talking a little bit about how evolution is applied to health sciences, we could step back to sort of a very basic application of evolutionary biology, which is to try and understand the phylogenetic position of various pathogens, to try to place them within the tree of all life, because when you have an unknown pathogen, you have no ability to predict its lifestyle. You have no ability to predict its susceptibility to different antibiotics or other treatments. And so a first step is trying to understand what group the pathogen is most closely related to, because that will tell you something about its natural history. It'll tell you about its particular adaptations. And so if we just look at this extremely simple sort of tree, just showing a sister relationship between eukarya, this is our group, and archaea, and an earlier branching for bacteria, I've just listed some of the different groups and the human ailment that they are causing. So there's fungi, which are responsible for various lung and skin infections, as well as allergies, flagellates, which cause diarrhea and sleeping sickness, amoeba, which cause diarrhea also, sporozoans, malaria and encephalitis, and microsporidia, also which cause diarrhea. Bacterial pathogens include spirochetes, responsible for syphilis and Lyme disease, and so on. Here in archaea, it's interesting, and I talked about these last time, methanogens that produce methane and can live in relatively methane-rich environments, halophiles, the salt levers, and other bacteria that live at extremely high temperatures and acidic conditions. We don't know there's no recognized pathogens for humans yet in those groups. I've presented some phylogenies, but I haven't told you yet, and so I'm going to do very quickly here how this sort of thing works. If you have a series of species 0 for outgroup, or O rather, A, B, C, and D, and then an understanding of the presence-absence of different characters. So here's 5 characters. Character 1 is present in species A, B, C, and D. Character 2 is present in B, C, and D, but absent in A, and so on. You can use this distribution of these traits of the organisms to build a hypothesis of relatedness. And there's 2 primary approaches. One is a maximum parsimony approach. This is really Occam's razor, just sort of transferred to phylogenetic analysis. You prefer the tree with the fewest number of steps, or a probabilistic approach, known as maximum likelihood. Building phylogenetic trees for a lot of these pathogens and their hosts has some real applications for understanding the origins of disease and how we might treat them. Here's a diagram with a phylogeny for a lot of immunodeficiency viruses from humans, the HIV, and from non-human primates. These are the SIVs. All the HIVs aren't each other's closest relatives. There's a cluster of HIVs up here, HIV-1, and another set, HIV-2s, down here. We can use a phylogenetic tree like this to understand that there's been 2 independent movements of an immunodeficiency virus from a non-human primate into humans in relatively recent times. This is a diagram which explains we can even hone that finer to understand the geographic region in which one of these transfer events first happened. So what we've got here is a phylogenetic analysis for different individuals from different populations of the chimp, Pan-troglodytes. So it's a phylogeny for chimps. In color here, we find that these are different populations which correspond with what are called different subspecies, Pan-troglodytes schweinfurthii, Pan-troglodytes troglodytes, and so on. When we do a phylogeny for their viruses, we can see that the viruses that are most closely related to those from humans that are right here, HIV-1 group M and group N, are these 3 which map to the range of Pan-troglodytes troglodytes. So the colors do correspond. And we can use this sort of inference to learn that indeed the outbreak really happened in this part of the world and was a transfer in relatively recent times from chimps to humans. So a phylogenetic and evolutionary approach can be quite helpful and informative. I've talked about HIV. It's spread and adapted to survive in humans worldwide just between 1950 and 1990. If you ever want to see evolution in the fast lane in observable time that you could just track by reading the New York Times, if anyone would say, well, we never see evolution happening, all you have to do is look at some of these viral pathogens and see how quickly adaptation happens, both by the pathogen and by the host. And I'll just show a series of slides for a variety of pathogens and make one or two comments quickly. Here's a similar slide for the smallpox virus. Earliest of infection of humans about 1100 BCE, so that'd be about 2100 plus years ago. It's adapted to thrive in dense human populations. It has evolved to find a evolutionarily successful strategy of making it in dense human populations. Its dispersal is by humans, by direct contact among humans or by contact with contaminated objects. Its recent evolution includes the notion that the last naturally occurring smallpox infection was in 1977 in Somalia. So it appears extinct outside of lab samples. So as far as minimizing the pathogen success in humans, vaccination is the primary way, even though we think it's gone from the wild. And so there's lots of debate about what to do as far as vaccination programs, because if it does come back, there will be a lot of susceptibility. Tuberculosis, like smallpox, is one that is deeply tied up with human history. And so I'll make this point here that the human cultural history with domesticating livestock means that there has been prior exposure to some of the microbes in some human populations before there's been exposure and evolution of immunity to those microbes in other human populations. Smallpox, of course, in the previous slide, humans from Africa and Europe that had prior experience with raising cattle, had some experience with cowpox, developed some immunity, and had better immunity than people in the New World had a chance to develop. And so in the case of the New World natives here, indigenous peoples in the New World suffered lots of mortality prior to any face-to-face combats with Old World colonists. And it's a somewhat similar story for tuberculosis. We used to think that this was involved in a similar way and came from the Old World to the New, with people who had some prior immunity to tuberculosis infections, but there's been some recent discoveries of tuberculosis in humans from about 4,000 years ago in South America. So it seems it has a much earlier history than we had thought just 5 or 6 years ago. West Nile virus, also much in the news. Having a phylogeny such as this was very useful in first understanding what was going on when there were a number of birds that were turning up dead around the Bronx Zoo in 1999. It could quickly be seen through a phylogenetic analysis that in fact it was closely related to yellow fever virus, that this was a flavivirus. There is now work on possibly developing a vaccine by starting with a vaccine from the yellow fever virus. All of this is possible because we know something about its phylogenetic history. The Ebola virus, this is one you've likely heard about. Our understanding is far from complete about Ebola virus, and we don't really know what species is the likely reservoir species where this virus occurs naturally. It appears to be in Central Africa based on the location of the outbreaks and phylogenetically related viruses, but we still don't know exactly who the natural host of this particular pathogen is. Influenza A has also been much in the news. Minimizing the pathogen's success, evolutionary success in humans, includes enhanced surveillance. And there's a lot of efforts going on, even now, this spring and this summer, on doing enhanced surveillance for avian influenza virus, which is a close relative of those shown here. And to attempt to minimize opportunities for virus mixing and transmission among host species, as well as the development of vaccines. So this is one that's strong on the radar screen for people interested in public health. And anthrax I won't talk about too much right now. Okay, here is our home, our planet, a place of limited resources. What we once thought were unlimited, but are limited to some degree. And so I want to transition a little bit here and talk about applications of evolution to conservation biology. Here are some mammal species. Here's a humpback whale and a calf. These are protected, so these are threatened and or endangered in different parts of their range. Here is a fin whale in full feeding mode, also protected. This is Hector's dolphin, also protected. And here's a minke whale, which are protected in some parts of their range and available for hunting in other parts of their range. Here's a minke whale being hauled up alongside a research vessel. If you look up here, it says research vessel. And they take, lots and lots of minke whales. Killing minke whales does have an effect on the ecosystems because killer whales, which ordinarily prey on minke whales, have less minke whales to feed on, so the killer whales then shift in their predatory habits onto sea lions and sea otters. And so conservation of some of these species does have broad consequences for the way that ecosystems work and ecosystems' health. It's also true that in a lot of whaling and fishing operations, you get unintended species that are taken in these large purse nets that are used in some cases even for whaling. So more dolphins are being killed and sold as whale meat. So it's legal to sell whale meat in some places, but not dolphins. And how would you know what you're eating? This looks really good. For many of you that eat a lot of stuff like that, you probably can identify what that is. Looks to me like tuna. But how do you identify it in a situation like this? Where things are frozen, or if you're a buyer and you're buying big containers, there's an identification problem. And how about this set of meat here? What is that exactly? And even in cases like this where there is no tag, here are the kanji. My understanding is it says whale meat, but in other cases, who knows? Maybe that would be dolphin meat. In other cases, there may be no label at all. And so evolution can be applied to enforcement of conservation laws by helping to identify products at fish markets and in meat markets. Well, how does this work? Well, you get a lot of samples, some samples of known identity and samples that you just go to a fish market or a meat market and you buy these samples and you plug them in to a phylogenetic analysis such as this. So here I'm showing a position of a minke whale, a humpback whale, a fin whale, Hector's dolphin, Commerson's dolphin, a killer whale, and a horse. And you can see the distribution of samples here. So this is illegal. These are illegal. These are dolphin samples. And so this is based upon some real analyses of samples that were purchased on the open market. This says horse down here. So there was even one sample that was labeled as something else that turned out to be some horse meat. By having a phylogenetic analysis, you can identify what these items really are and then hope to enforce conservation laws. And in fact, this is being used fairly routinely now. So that's a way in which evolutionary biology can be applied to conservation of natural resources. It also can be applied to sturgeon because caviar, that is the eggs from some sturgeon, are legal and they're illegal from other species of sturgeon. And even in falconry trade. by trying to identify the geographic origin of particular gyrfalcons. So you can build a phylogenetic analysis like this for different geographic regions and find out if some falconer's gyrfalcon is from an area where it's legal to collect them or not. Evolutionary biology is also applied in some basic conservation ways and just identifying what taxa are out there to be conserved. We still don't have a very good handle on the diversity of life forms. This slide was taken from a textbook, and I noticed that here are animals and here are insects. Last time I checked, insects were animals. But I guess they wanted to show here that, in fact, there's a very large diversity of insects. These are the numbers of named species, and these are gross underestimates, of course. But they do give you an idea of what we currently have as far as named taxa that we might be concerned with preserving the diversity of these taxa and the diversity that's present within their genomes. And we still have a lot to do in terms of identifying these unique taxa, and an evolutionary approach is going to be very important in identifying the different lineages in the tree of life for conservation purposes. And here's a brief schematic. So systematists that work on developing phylogenetic hypotheses identify diverse evolutionary groups arriving to the present from the past. So we look at the diversification of lineages within parts of the tree of life. So all of these are things that evolutionary biologists would do. During the present, we can do ecological studies to show the life support systems for biota in the present, understand how ecosystems work. And in the future, evolutionary biologists can seek to ensure diversity and the functionality of ecosystems into the future. There's lots of discussion about maintaining ecosystems services because they are so fundamentally important to continued human health and human welfare. Having clean water, having air, since plants and bacteria do such a great job in providing oxygen. We really are tightly linked. The field of evolutionary computation and the development of evolutionary algorithms. And these are used to emulate processes of evolution to solve real-world problems in engineering, industry, and science. And this is applied evolution, I would argue. How is it applied? Well, perhaps you are being charged at work with the need to optimize the orbits of groups of satellites used in communications, industry, espionage, and defense to maximize coverage of the Earth. Because if this is the Earth and you've got a satellite going above, there's going to be a lot of the Earth that's in the shadow. The satellite cannot directly receive or send signals. And so you've got a series of satellites, but how do you regulate their orbit to minimize the amount of blackout? That's a complex problem and an interesting problem, and you could use an evolutionary computation approach. You may have to consider scheduling airline landings, departures, baggage routing, security clearance for airports globally. A hugely complex problem. You might even have to schedule the room usage at the University of Michigan for who can use what room for what exams when. And they are using, there are some commercially available evolutionary algorithms that are used for solving problems like that. Also, routing of data in telecommunications networks is a place where evolutionary algorithms can be used. Predicting currency rates, stock picking, and also playing chess, of course, are places for applications. So how does it work? What is it about these algorithms that's evolutionary? So I'll outline 5 steps. First, because evolution is something that happens in populations of organisms, you take a population of candidate solutions. You need a whole series of possible solutions. And then you take a subset of relatively fit or high-performing individuals, and you select those possible solutions as parents. Those parents are subjected to mutational changes. So these are just solutions of some patterns you're looking for. You start using random variables, and you just change those patterns. You change the way the satellites are going to orbit. So you subject them to mutational change. Then you get the altered candidate solutions, the progeny. These comprise a new population, and if the optimality criteria for termination has been reached with the current progeny, the process is stopped. If not, you begin again with step number 2. In hindsight, it all seems so obvious, but this hadn't been tried until relatively recently, and these approaches vastly outperform a human, as sophisticated as the human brain is, just a human trying to work things out with a pencil and paper. You just start mutating possible solutions and then go through lots of bouts of mutating those possible solutions and then measuring their performance. Okay. Let me shift now to talking a little bit about applications of evolutionary biology to some aspects of human culture. And in doing this, I want to stress the point that human cultural evolution, or the evolution of cultural phenomenon, it evolves in a metaphorical sense. Okay, the capacity for human cultural development evolves together with human anatomy and physiology, with brain form and function, with our mental acuity, and with aspects of social behavior. However, the evolution for particular features of human culture, such as language and religion, remains a metaphor. Why do I bother to make this distinction? And that is because it's important in understanding the mechanisms. The distinction between biological evolution and cultural evolution as a metaphor is important for understanding the ways in which they happen. So that's sort of a caveat to what I'm going to do now, which is to present some ideas that are largely fun, I think, but do show that there are some applications of evolutionary biology and evolutionary methods, particularly phylogenetic analysis, to understanding some of the change over time of human cultural phenomena such as language and religion. So etymology is the study, of course, of the history and origin of words. And here's a picture from the famous Monty Python skit, which is the result. As a result, we now have the word spam. And here, Mr. and Mrs. Bunn are at their table in the cafe asking what's for breakfast, and the waitress is telling them, you can have eggs and bacon, egg, sausage, and bacon, egg and spam, egg, bacon, and spam, sausage and spam, spam, bacon, sausage, spam, spam. And suddenly, these wannabe Vikings start singing about spam, spam, lovely spam, lovely spam. And as in typical Monty Python sort of theater of the absurd way, this spam takes over this whole conversation until the waitress yells out, "Shut up," to these Vikings. And this humorous skit appealed to some of the early internet users who were developing some of the algorithms for spam as signifying something that was sort of unwanted junk that just kept cropping up. And the notion of spam as unwanted internet mail was born. So this is etymology. And so the basic idea is that as a metaphor, words evolve, languages evolve, and of course our books evolve, and the ways that historical documents will evolve, and I'll talk a little bit about that. Here's a sampling of 3 different words in a bunch of different languages. If we just look at fish, poisson, fish, pescado, fisco, fisc, Pisces. And so from English to French, German, Italian, Spanish, Dutch, Esperanto, Swedish, Latin. There was a guy named Morris Swadesh who, in 1952, developed this approach to looking at the relatedness of languages. You simply developed tables like this, and you looked for cognate words, words which were similar in the way they sounded and the way they were spelled and what they meant. And then if you get enough of these, you could begin to understand in some way the history of cultural change for language. That was really the start, and things have gotten much more sophisticated since then. And here's a phylogenetic analysis using a maximum likelihood approach for some languages, using lots and lots of words, tens of thousands of different words. And it's even possible to try to estimate dates. I won't try to defend these as reasonable, but there are the beginnings of estimating dates for when different languages began to diverge from each other. And so here we see that in this evolutionary analysis, using maximum likelihood statistics, based upon shared words, what are thought to be cognate words, show that Irish and Welsh are sister languages, no surprise there, that diverged about 2,900 years ago. And here's a split between Dutch and English versus Swedish and Danish about 1,750 years ago, and so on. So we can use an evolutionary approach to try to understand the history of language, which is one of our most important cultural aspects. We also know that there is lots of borrowing between languages, just as there is that we see now at the molecular level for genomes of organisms. So here is a phylogeny for biological organisms, and here is a phylogeny for languages. And in each case, this is a horizontal transfer event. So I'll just focus on this one about languages here. And for English, isn't it interesting that we have both beef and cow, pork and swine, mutton and sheep? Why is that? Well, English is close to German. So we have cow in English and Koch, I don't know the correct pronunciations in German, swine and Schwein, sheep and Schaf. But we also have the French, beef and boeuf, pork, mutton, and so on. So there are analogies, at least. This is a diagram just showing part of the history of translations of the Bible. So we can have metaphorical evolution of words, of languages, and of course documents that have long been held by humans as culturally important, and they do change over time, especially when they go through a series of translations. So here's a diagram showing part of the history of the Bible, starting with Hebrew scriptures down here, through a whole series of translations, here from Hebrew scriptures into Greek in the Septuagint, beginning about 275 BCE, before the Common Era. Here is the Greek New Testament added to scriptures, this beginning about in the year 100. Here is the Vulgate, written about 400 in Latin, and then moving from Latin into human. Here is Martin Luther's translation in 1522, William Tyndale's translation in 1526, and so on. Here's the King James Version. So we can use sort of an evolutionary approach to look at the history of some of these culturally important documents. And it's kind of fun to look at it in the sort of tree diagramming. And of course, there are lots of horizontal connections here because there is a great deal of borrowing across cultures. So I don't mean to imply this is a strict genealogy, but there's certainly historical connections that can be looked at and understood here. And so to give you a sense of this change in this document over time, let's consider a well-known passage from the King James Version. So remember, this was translated at the request of King James to get rid of a lot of marginalia that had been written by people that were giving King James political troubles. He asked for a translation that was free from all this marginalia, and in some ways it helped him, in other ways it didn't. But anyway, from 1611, the Lord is my shepherd, I shall not want. He maketh me to lie down in green pastures. He leadeth me beside the still waters. He restoreth my soul. He leadeth me in paths of righteousness for his name's sake. Yea, though I walk through the valley of the shadow of death, I will fear no evil, for thou art with me, thy rod and thy staff, they comfort me. 1611. Here is a translation that was published in 1985 by the New Jewish Publication Society, based upon new scholarship, based upon new archaeological findings and new understandings of linguistics for Hebrew and Aramaic both, and trying to do as accurate as possible a translation. And it's a little bit different. The Lord is my shepherd, I lack nothing. And this was done in modern idiom, English idiom. He makes me lie down in green pastures. He leads me to water and places of repose. He renews my life. He guides me in right paths as befits his name. Though I walk through a valley of deepest darkness, nothing about death here, I fear no harm, for you are with me. Your rod and your staff, they comfort me. Things change. Sometimes it's subtle, sometimes it's not. Consider the earliest translation into English by Wycliffe in 1384. And recall, too, that in doing this, so he was working on this translation into English for the first time say from Latin. And for this service, he lost his teaching position at Oxford. He had to flee to Germany, where he was working in secret to do this publication into English. And 40 years after his death, his bones were dug up and burned on orders of the Pope. So, we think we have troubles now. I mean, people have long had troubles when you're dealing with sort of a volatile, culturally important document such as this, and it does change over time. So it's metaphorically evolutionary, and we're interested in history, and we can recover that. And just to get a sense of the changes in syntax, you can look at some of this. Written in 1384, "The Lord governeth me, and no thing to me shall lack. In the place of leisure where he me full set, or water of fulfilling he nourished me, my soul he connower tide. He brought down me upon the skies of righteousness for his name. For we, and if I shall go in the middle of the shadow of death, I shall not dreaden Euless, for thou art with me, thy guard and thy staff, thou hand comforted me." Forgive my pronunciation. So, things do change over time, and they have important consequences for human cultural evolution. There is a statement in the Old Testament, Isaiah 7:14. The prophet Isaiah says to King Ahaz of Judah, "Behold, Ha'alma," that's the Hebrew, "shall conceive and bear a son." In the New Testament, in the Gospel of Matthew, this prophecy is restated. "Behold, a virgin shall be with child and shall bring forth a son." Ha'alma was translated as virgin. So the Alma was translated to the Greek Parthenos, meaning physical virgin. Alma is accurately translated as young woman, with no implications about virginity. It was just a young woman, robust in the prime of life. That translational change has had a big impact on the canon and on the cultural history for human beings. And it has to do with the evolutionary change of languages and documents that are written in these languages. And languages fall in and out of favor if you look on a large enough time scale. What about the religious traditions themselves? Here is a picture that was taken in 1893 of some Jews praying on the Mount of Olives in Jerusalem. Jews have been interested in being buried there since biblical times because it's said that the redemption by the Lord would begin in this area, on this particular slope. In the background, you can see the wall around the old city of Jerusalem, built by the Turks in the 16th century. You can see the Dome of the Rock, built by the Muslims in the 7th century, and according to tradition is where Muhammad ascended to the heavens and to God. And the Dome of Rock itself is built on top of the site revered by Jews and Christians as the Temple Mount, first constructed by Solomon about 3,000 years ago and rebuilt in the year 70. And tradition there holds that Abraham prepared to sacrifice his son Isaac on that spot. So there's a lot of confluence here. There's a lot of ongoing tension over control of this area. It's very important to, in the context of three primary human cultures, Judaism, Christianity, and Islam. And here's a more recent picture taken. You can still see the Dome of the Rock and the walls built by the Turks and some additions to the landscape. And of course, the cultural contest continues. And so what can evolution tell us about all of this? And my basic point here is that there is it is a single family in a lot of ways, even for these cultural institutions of these traditions. So if we start with a sort of a phylogenetic approach, and let me go ahead and build the tree right before your very eyes. And this is a very rough estimate of an analogous evolutionary tree with some characters on it. The first split is between these cultural traditions as a single group, a set of related traditions, and other groups. And one of the first things we can note is that circumcision has been around a long time and is not anything that defines any of those particular groups. There are lots of human traditions outside of these that have circumcision as an important part of their culture, both male circumcision and female circumcision, in fact. Monotheism, sort of interest in these prophets, dietary laws, Sabbath on the Saturday, and holding of Jerusalem as a holy site is something shared by all of these traditions historically. These dark rectangles mean the origin of a trait. Where it's a light rectangle filled in with some dots, that means that trait is lost. So following this metaphorical split here, this is a Christianity lineage. Jesus becomes important, died in about the year 29 of the Common Era. The circumcision trait was lifted, not necessary. Dietary laws, not necessary. I suppose both of those help a religion spread. Hi, I've got a new religion for you. The first thing we want to do is circumcise you and talk about what you can't eat. And so it makes sense to sort of relieve those things if you're trying to spread a new religion. The Sabbath was changed to Sunday. Rome took on a more important role compared to Jerusalem, so that's why I said it as a character loss here. Islam, Muhammad is the primary prophet there, died in the year 632 CE. Also Sabbath on the Sunday. Mecca has more importance. And here I have Judaism and the Talmud within the Judaism, the tradition of all the laws that are bound up in the writings of the Talmud are relatively recent, and in fact, they post-date Judaism as practiced by Jews in Ethiopia, most of whom have now been relocated to Israel. But anyway, it's possible to take an evolutionary approach to try to understand some of these cultural traditions, and I'll just show you, I think, just a few more to give you an idea that I think in the future there will be more work done on looking at some of the relatedness of these traditions in an evolutionary context. And so the worm is turning here. That's what I'm trying to do. Evolution can be used to learn about religion in the sense of it being the history of human cultural development. And there may be a time when it's possible to do more quantitative analyses on these different religions. So here is just a series of differentiation for different cultures within Judaism, starting with the ancient Israelite tradition. An early difference arose between the Samaritans. There are still a small group of Samaritans practicing what's close to an ancient Israelite religion and then ancient Judaism and so on. So we can recognize some different traditions there. Here's a tree for different groups of within the Christian tradition. An early split can be recognized between the Western Church and Roman Catholicism and the Eastern Church. And the last tree I'll show is for Islam. The primary divergence here would be between the Shia tradition and the Sunni tradition, and this initial split occurred over what else, as happens not just here but everywhere, a dispute over who has authority to rule. So the more things change, the more they stay the same. And it's interesting to note that here is Baha'i, which has its roots in Islam. And of course, Baha'i embraces all the prophets from Abraham and Moses through Jesus, Buddha, Krishna, Zoroaster, and some others. And it actually incorporates both religious and scientific views and attempts to give scientific views primacy. So anyway, there are some things that can be done to look at the evolution of human cultures. And I'm going to stop there, and thank you for your patience.
Saturday Morning Physics is sponsored by the Physics Department of the University of Michigan, by the Dr. Remlo Tiffany Endowment, and by gifts from friends of the program.